Is Ashley Lee, and I am recording myself for IL 650, my tech connected lesson. So actually do still teach a class. I'm the curriculum coordinator at for Jackson County Schools for math and science, but I actually still do teach a class. I teach a dual enrollment class or an advanced class at one of our local high schools, and I wanted to show you how I do that. So I pre-record everything I do just because I don't know what's going on. I'm not physically there at the high school with them. I might be in a meeting. They might have an assembly or something like that. So I pre-record everything, and the facilitator does the to-do list for that day. And so this has been sent to the students. So the first part of the lesson is our bell ringer, and it's called Prime Time. I got this idea off the internet. Instead of just doing a regular bell ringer, we have a Prime Time notebook, composition book. I almost forgot the name of it. We do a composition book of all of our prime times, and they turn it into that facilitating teacher every day, and they get it, and at the end of the week I will go get them, grade them. Sometimes I might pop in or have her look through them for me and kind of give me a gist for the next day. So this lesson is about operons and gene regulation. You can see that on the form that I turned in my lesson plan. But it says, how do eukaryotic cells regulate their genes? That come from the previous lesson. What differentiates a prokaryotic cell from a eukaryotic cell? So that's some prior knowledge that they need to know, and it's from a previous chapter as well. And then it says, what do you know about prokaryotic gene regulation? So this is kind of a peek into what we're going to talk about today throughout this lesson, and the students have a little bit of prior knowledge because they have looked at their Quizlet, which has all the key terms, and they have read their chapter. So this is the first thing they do. It's on the board with the facilitator when they come in the room. And after that, they go to the introduction video, and I just wanted to kind of show you some of this, and you can see what I use for my introduction videos. I love Amoeba Sisters. They're great. They're fun to listen to, and it just kind of grabs the students' attention.
Have you ever thought about what a disaster it would be if the cells in your eyes started producing the same hydrochloric acid that is made by your stomach cells? Your stomach cells produce hydrochloric acid to help break down food, but you definitely don't want that in your eyes. Thank goodness that doesn't happen. But it's surprising because both your eye cells and your stomach cells contain all of your DNA. Your DNA, all of it, is found in your body cells. But see, the portions that are used need to be regulated somehow. Otherwise, we could end up with something ridiculous, like eye cells producing hydrochloric acid. And that wouldn't just be a waste of resources, that would actually be very difficult to explain to your friends. You want genes to be regulated, controlled. Remember that genes are made up of DNA. DNA is used to give instructions for the production of proteins in the process of protein synthesis. But an important concept is that there needs to be a method of determining which genes will be turned on and which genes will be turned off. This is called gene regulation. And there are many ways that genes are regulated. In your human body cells, you can have proteins that can bind to certain gene regions to increase the rate of transcription for the transcription enzyme RNA polymerase. Or you can have proteins decrease transcription to the point that it may not be transcribed at all. This is a form of gene regulation. Your eye cells don't use the portion of DNA that codes for making hydrochloric acid like your stomach cells do, because there is regulation in your cells to determine which portions of the...
So I just want to note that this is prior knowledge. This is the first portion of the chapter. Once we get into the latter part of the video, it talks about what we're going to learn about with this lesson. I'll kind of skip over and let you see that.
This is an example called a lac operon. Notice there's a promoter region of the DNA, the operator region of the DNA, and there are 3 genes here that code for enzymes that help in the process of breaking down lactose. Lactose is a sugar. If lactose sugar is around...
So you can see that the Amoeba Sisters do a great job of creating their cartoons and making it fun for the students to learn. I always love to show them at the beginning of my lessons if I can, just to kind of grab the students' attention and make it more almost relatable. If they can visualize it like this, I think it's even better. So sometimes I use Edpuzzle with these because I'm not there. Edpuzzle, the program or the app Edpuzzle, ensures that the students are actually watching the video, and if they get on, like if they went to another tab, it'll stop. So that's good. They can't, try to look up something else or something like that during the video. It also has questions on the video, so they are questioned throughout the throughout the video, the intro video, to make sure they're paying attention and they understand. But I didn't do this today, just to show you the video I thought would suffice. So the meat of the lesson for this lesson on operons is used with an annotated note that I did. I used the app Notability, and this is something that I link to the students, and they watch it as a class. The facilitator will put it up for me, and they watch it as a class, and I wanted to show you some of this. Okay. I also want to note that they have their own paper and they follow along and write as I write. Hey class, so you should have done your prime time this morning, and then you watched our introduction video about gene regulation in prokaryotic cells. So now we're going to do our main activity for the day. So again, you guys know I'm using Notability. I love it. I hope it works out for us today too. Hope there's no kinks. All right, so you should have this on your desk. You have a printout version of this, and you should be able to annotate this as we go with me, okay? So it says prokaryotic gene regulation. Prokaryotes and eukaryotes both contain chromosomes. However, their chromosomes are organized differently, and that was one of your primetime questions. Prokaryotes have a single large blank chromosome, while eukaryotes have multiple blank chromosomes wrapped around each histone protein, okay? So the first blank is talking about prokaryotes and that what kind of single large chromosome do they have? They have, so let's just look right here. So this one is a eukaryotic piece of chromosome, and this is a prokaryote, okay, it's chromosome. So it's a circular, if you want to write circular chromosome, while eukaryotes have multiple linear chromosomes wrapped around those histone proteins that we've learned about. Okay, so let's read on. It says eukaryotes have or use transcription factors to regulate gene expression. Okay, so we've already kind of covered that, but I'm going to highlight it just in case. Make sure you know that. Highlight it on your paper. And it says prokaryotes use operons, and that's what we're going to learn about or review today, to regulate their gene expression. So let's learn about some operons. You should have already read your chapter. You should have some background knowledge of what an operon is. So it says an operon consists of a promoter, a blank, and the genes they control. So in the blank, we have to write the last thing that's missing that I haven't highlighted, which is an operator. Okay. They have 3 parts. If you'll take note, there's also another part out here to the left of the operon, okay? We'll learn about that. So the students have to watch that. They have to annotate their own notes, and then they have to actually make them upload them or take a picture of them and just upload them for credit so I can make sure that they're doing it. It's just accountability. But I do let them keep these because I think they're a great little, almost like a graphic organizer for them to use to study. Okay, so the next part of the lesson after we do this is I have them for 90-something minutes. I think it's 93 minutes maybe that they're in the class. I have them for a pretty good bit. So at this point, they usually will take a break, and then they'll come back and do their end of lesson assignments or assessments and be done for the day. So this is the next portion of the lesson. This is the closing or the summarizing portion of my lesson plan. This is something that I did not create myself. I found it and reused it. That's what we do in education, right? We just borrow and steal and make it our own. So I found this. I loved it. I love that it's interactive, that the students, that it uses technology because I am teaching virtually. It helps me so much, but it still allows the students to do something a little bit different, and I appreciate that. So what the students do, they've already learned about their inducible and repressible operons. They already have their little graphic organizing annotated notes to help answer this with. And what they will do is they will just read, decide if the statement is describing what type of cell, and they will drag and drop for that. Again, this is another portion. They will take these little squares and they will drag and drop for in the options as well as the names of the parts of the operon. So I thought this was really cool. It's just a different way to learn. And again, it goes all the way down. It follows the same diagram, almost even sometimes the same shapes as their notes do. So I thought this was a really good assignment for them. It even goes in detail about the different types of operons. And once they finish, they will go to our Schoology, which is our LMS, our learning management system for Jackson County Schools, and they will post them to the assignments for a grade. So I wanted to show you this too. This is almost like the base or the hub for all of our information because I am in a different location than them. Every week I will post, I usually do it on Sundays, I will post almost like an agenda for the week. And again, here is this agenda that they did this on. And it just kind of helps the facilitator. She goes over it every day and says, this is what we're doing today, so the students know. It's almost like putting their objective on the board. And this is where they will turn in their annotated notes and their drag and drop. The last thing that they do is an exit slip, and they'll just make them, it's on the last part of that drag and drop. It tells them what to do. They just grab a sheet of paper and they draw their own operon and label it, and they turn it in as they leave. So again, that helps me. That's another way to formally assess if they got the lesson today or if we need to re-review. And I think that's it. If you have any questions, you can email me, and I'll be happy to answer anything. Thank you.